We're about to announce the 13th Annual William Hancock Award. This is really an award to recognize the outstanding and sustained work in regulatory science. And this year, I'm going to ask a fellow board member, Sarah Kennett, to come up and announce this year's awardee.
Thank you, Jamie, for sharing the opportunity to present this year's Hancock Award, with which we're honoring someone with whom I had the privilege of working for many years. So yes, clue number one, this year's winner worked at the U.S. Food and Drug Administration for more than 30 years. And it's therefore even more disappointing that our FDA colleagues aren't here with us to help celebrate this year's honoree. However, we're very fortunate to have so many of you from our CASSS community here at this year's WCBP, because this person has also been an active part of the CASSS community for as long as there has been a WCBP. The CASSS Board of Directors has selected Dr. Marjorie Shapiro to receive the William S. Hancock Award for Outstanding Achievements in Regulatory Science, CMC Regulatory Science. Margie recently retired from the FDA after 31 years of service to the agency and more than 34 years of service to the U.S. public. For 20 of those years, Margie served as chief of the Laboratory of Molecular and Developmental Immunology within the Office of Biotechnology Products. Margie led the only FDA research program with a direct focus on antibodies and oversaw a broader laboratory group studying mission-critical topics ranging from immune system effector function-based mechanisms of action to viruses and prions. In her capacity as a reviewer and researcher at the FDA, she contributed to the review of thousands of INDs, BLAs, NDAs, PMAs, MFs, and the subsequent amendments and supplements, ensuring the quality of biotech products. She was a reviewer or lead for many of the FDA approval firsts, including the first radioimmunotherapeutic approved for treatment of cancer, the first antibody-drug conjugate, and the first protein therapeutic claiming a manufacturing design space. Margie's broad experience and expertise, along with her ability to follow and apply new scientific knowledge, led to her involvement in many advancements in the growing and ever-transitioning fields of biotechnology and regulatory science. Margie led or was involved in the development of the majority of FDA's antibody and other therapeutic protein-related guidances, from the 1997 Points to Consider, to transgenic plants, to biosimilars, to COVID-19 related documents. She played important roles in developing and communicating policy on many precedent-setting topics, emerging complex issues regarding novel products, processes, analytics, and regulatory pathways. This extended outside the walls of the FDA. Margie's worked with USAN on antibody nomenclature, has presented Antibody 101 to U.S. Department of Justice lawyers, and served as a member of the NIH Study Section for Small Business Innovative Research and Small Business Technology Transfer Grants. She's well known globally for her major contributions in the regulation of antibody-drug conjugates, complex antibody-related formats, and biosimilars. During her time at FDA, Margie influenced policies and practices in support of risk-based, fit-for-purpose, and state-of-the-art concepts. Importantly, in addition to communication through guidance, she frequently spoke both internally to FDA and globally, and also authored publications to not only present, but also explain policies and expectations. She reached out and listened to regulators internally and externally, and industry large and small. This listening and learning is a hallmark of Marjorie Shapiro. Wanting to understand what's happening in all aspects of our industry, considering how industry and regulatory science is progressing, and using that to become a mentor of better regulators. Margie's been a supporter and active member of the CASSS community since the first WCBP conference, where she presented on generic and modular viral clearance, one of the many novel concepts she helped develop during her time at FDA. She served as the FDA co-chair of WCBP for 5 years, and for the 10 years since has been a member of the WCBP Steering Committee. She also co-chaired 7 WCBP plenary sessions and 2 workshops. She presented talks and sat on panels at WCBP, CMC Strategy Forum North America, CMC Strategy Forum Japan, Bioassays and Midwest Discussion Group, the Northern California Discussion Group, and even an early CASSS webinar. And critically for all of us, Margie's been a very active participant during panel discussions and workshops, voicing thoughts and asking and answering questions from the podium and the audience. Beyond CASSS, Margie has lectured at the University of Maryland, Baltimore County, and Georgetown University. She's provided training through the FDA Small Business Regulatory Education for Industry and different associations. She's been a moderator for conferences with ISPE and New York Academy of Sciences, and she's presented talks at a very large number of additional global conferences. And one of the best things about Margie's talks are their practicality. Right? Who can forget that highly informative pet peeves talk that Margie presented or co-presented at WCBP and other venues? And it's not only in such talks that Margie is practical and giving. Margie helped develop a generation of reviewers and colleagues in industry. She always shared her time with everyone, the lab-based staff, reviewers, industry. Whether it was years of training for individuals directly under her supervision, bits and pieces of mentoring or coaching, a quick consult regarding a specific issue, or working with industry counterparts on strategies for their products, she was there for us. Hers was the door I knocked on at 7 a.m. to seek guidance, someone I could count on to help set me straight or do critical reviews of my own reviews or draft documents and communications to industry, someone to challenge my thinking and expand my views. And she's been that someone for so many people. Because her words are spoken with knowledge, science, and reason behind them, her voice carries weight and is well respected within and outside the walls of FDA. Someone else recently said, when Margie talks, people listen. And we on the board decided that they do so for good reason. And so on behalf of the CASSS board, and with thanks from CASSS, our industry as a whole, and all the patients we teach, I present to Marjorie Shapiro this physical commemoration of the Hancock Award, and congratulate her on an impactful career. Thank you, Margie.
Wow, thank you, Sarah. That was quite a lot. I'm completely overwhelmed. I want to thank Sarah for the introduction and CASSS and the CASSS board for this incredible honor. Last fall, when Mark Shennerman said he wanted to talk to me about CASSS, never in a million years did I think he was going to tell me that I was receiving the William S. Hancock Award. I just always thought of myself as that crazy person at the FDA who loves antibodies more than anyone else. So let me explain how that came to be. Before I went to graduate school, I worked for 4 years for Mike Potter. Mike Potter was an investigator at the National Cancer Institute who developed the mouse plasmacytoma model, which is the mouse version of multiple myeloma. And one of the questions he wanted to answer was, what are myeloma proteins? Well, myeloma proteins are monoclonal antibodies, but back then it wasn't known for sure if they were representative of natural or normal antibodies. So we purified the antibodies from ascites, starting with ammonia sulfate cuts, size exclusion chromatography, and DEAE anion exchange chromatography. Most of the antibodies were IgA, some were IgMs, but several were IgGs. So we were very happy when protein A came around because it made the purification that much easier. We also characterized these monoclonal antibodies by screening them against a panel of antigens using classical immunochemical techniques such as octolony and hemagglutination inhibition. And I imagine there's some of you in here who've never heard of those. And we isotyped them using agarose gel electrophoresis. I had the opportunity to modernize the lab by bringing the technique of radioimmunoassays into the lab by visiting a former grad student of Dr. Potter's at the University of Pennsylvania. We also isolated heavy and light chains and prepared to sequence them using old-fashioned Edmond degradation, and I had to learn to de-block the heavy and light chains that had an amino-terminal pyroglutamic acid residue. And then we also used some of the isolated heavy and light chains to make anti-idiotype anti-sera. So my love of antibodies came from Mike Potter, who was enthusiastic about everything. He would take me to various labs across the NIH campus to introduce me to other immunologists and to learn various technologies. But he never used elevators, and so we would often walk up to the 11th floor of the clinical center, which housed the lab for immunology. Mike Potter won several awards for his pioneering work with plasma cells, but importantly, he believed that science was driven by curiosity, not competition, that science should be an open pursuit with national and international colleagues, sharing ideas, techniques, and importantly, materials. He organized an annual conference called the Homogeneous Immunoglobulin Workshop to talk about the current understanding of antibodies, and I was fortunate enough to meet many of these pioneering antibody scientists. One of my jobs was the curator of the ascites panel. Hybridoma technology didn't exist yet, and scientists were interested in testing the plasmacytomas to see if they had specificity against the antigen they were studying. So I would send about 150 microliters of 100 different ascites samples. And one of the people who I gave samples to was Katie Stein, who would eventually move to CBER and become the founding director of the Division of Monoclonal Antibodies and was the 2023 Hancock Award winner. I then went off to graduate school, got my PhD studying the mechanisms of VDJ recombination, and several years later as a postdoc back at NIH, I ran into Katie. This was just after PDUFA 1 was passed, and CBER reorganized to create the Office of Therapeutics Research and Review, which included the newly formed Division of Monoclonal Antibodies. She recruited me to join, and it was a good home for me. It wasn't long before I could see the connection between Mike Potter's generosity and the development of therapeutic monoclonal antibodies. Through sharing knowledge and materials, hybridoma technology was developed, antibody promoters enhancers were discovered, chimeric antibodies were generated, and all this knowledge was applied to develop therapeutic monoclonal antibodies. Some of the grad students and postdocs in those labs of the scientists who participated in homogeneous immunoglobulin workshops or who got materials from us, or the subsequent generation of grad students and postdocs, went on to work in the biotech industry, developing the first generation humanization processes and later humanized mice. In the Division of Monoclonal Antibodies, I had many opportunities to be involved in reviewing antibodies with novel issues and policy development around these issues. And I learned two lessons early on that can be illustrated in this slide by my attendance at the first WCBP meeting in 1997. The first lesson I learned is that regulators need to always be open to advances in technologies, but that sponsors need to provide rigorous data so that we can make scientifically sound decisions. I was assigned to review two submissions containing data on virus clearance studies from two sets of antibodies targeting different tumor antigens. The sponsor didn't want to have to do the virus clearance studies on the full set of virus clearance studies on about 10 antibodies. So based on Katie's advice, they provided data from different purification steps for different antibodies using the same or slightly different purification processes. Upon review of the data and internal discussions, we determined that the data supported the request, and this led to the development of the policy for generic and modular virus clearance studies, which was published in the 1997 Points to Consider Antibody Guidance document that I got to present at the first WCBP. The second lesson I learned at this conference came from meeting the CMC Regulatory Affairs person from the company that submitted the first commercial IND I was assigned to review. The IND was so interesting, and I was learning so much about how antibodies were designed, developed, manufactured, tested. I had a lot of questions. Many were for curiosity, and the sponsor also submitted information about the facility, which I had no idea how to review. So Katie suggested we get input from our facility experts. In the end, I sent about 30 to 35 questions for a phase 1 IND that did not go on hold. Some were curiosity types of questions, many were about the facility. The regulatory affairs person told me that the questions led to a lot of anxiety at the company, and a lot of people had to scramble to get the answers. I had no idea that such a junior reviewer at the FDA had that kind of power. So I realized then that no matter how curious I was and how much I wanted to understand things, if it wasn't important for the phase of development and from a regulatory standpoint, I should be mindful of the types of information requests I sent. So meetings like WCBP are where I ask my curiosity questions. And I would also not send those facility questions today, or if I did, it would have the comment like, these are for your consideration as product development progresses, and you don't need to respond to them. A different lesson about the power of the regulator came many years later. I was reviewing comparability data in a BLA supplement and wanted to chat with the sponsor about some of the data. The CMC Regulatory Affairs person set up a TCON for 4 p.m., except that he forgot about the difference in the time zones, the difference in time. So Keith Weber and I got on the call, and no one from the sponsor was there. The sponsor's rep was very upset that he made this mistake with the agency, and he was worried that it would impact the review. We reassured him it was not a big deal, and I said that I could easily have made the same mistake. We eventually had the discussion and resolved our concerns. After that, we were able to laugh about the mistake, but the lesson was to always take the job seriously, but not myself. If everyone is at ease, we can have better, more open discussions. Since that first WCBP, I've spoken at many CASSS and other meetings, usually with much more confidence than at that first presentation. I looked at several of those presentations and realized that my advice has been consistent over the years, that sponsors should be innovative, FDA should not be scared to agree to new ways of doing things, but it always comes down to doing good science and providing adequate data to convince us. As I said in a talk at the 2018 WCBP on state-of-the-art methods, we are professional worriers. Put yourself in our shoes. If you are proposing something new, what would be a regulator's concern? The other consistent theme across my talks has been that the quality of your submissions matters. In recent years, I've suggested that you think about your submissions as a kind of educational document, especially when a new technology is being introduced. I remember when anion exchange membranes were new and we didn't understand how they worked for virus clearance. Were they more like the chromatography resin or more like a membrane or a combination of both? The submissions didn't explain this, so many IRs were sent. It certainly would have helped if the explanation had been there. A final theme across some of my talks became more apparent the longer I worked, and that is that we are never done learning about our products, how our products work, and the diseases they're used to treat. So we need to continue to be curious and open to new ideas, that conventional wisdom is sometimes wrong, and figure out how health authorities and industry can work together to apply new knowledge for both old and new products to ensure that patients can continue to get safe and effective medicines. So I'm grateful for the many opportunities I had to learn about the development, manufacturing, testing, and mechanisms of monoclonal antibodies, and I look forward to the continued approval of many more. And finally, I want to end on a more personal note. I grew up in this town and I'm a second-generation civil servant. My mom came first during World War II to work for the Navy, having the heavy burden of typing letters to families, letting them know that their son or husband wasn't coming home. My dad came later after a stint in the Army to work for the Office of Price Administration, and then after the war was career at the Department of Justice. And they were proud of their civil service and believed that what they did mattered. And they passed that along to me. So it was an honor and a privilege to be a civil servant and to do my part to help protect and promote the health of the citizens and the residents of the United States. Thank you again for this honor.